Dear students, I heartily welcome you in my channel, Doctor at Phytopathology. Actually, this channel is dedicated to those students who are pursuing their uh, graduation and post-graduation uh, studies in plant pathology or in agriculture. Actually, today we'll discuss very important topic, that is the phytoalexin. Actually, phytoalexins are very wonderful tool for the plant disease suppression and management. Let me know what are phytoalexins. Actually, phytoalexins are low molecular weight antimicrobial compounds that are synthesized de novo and accumulate in plants in response to infection or stress due to wounding, freezing, ultraviolet light exposure, and exposure to the microorganisms. So here students, de novo means new. As the definition of phytoalexin tells us that any stress, biological, chemical or physical stress, if a plant faces, he produces phytoalexin. Phytoalexins are antimicrobial in nature. They can inhibit the growth of fungal, bacterial, or other type of pathogens. And phytoalexins are involved in the complex of defense system that allows plant to manage invading microbes through uh, development of hypersensitive response. Let me know some facts about phytoalexin. Actually, the term phytoalexin was coined by Müller and Borger in 1941. Then in Müller in 1958 discovered first phytoalexin when he was working in bean uh, about the pathogenesis of Mononella fracticola, a pathogenic fungi. So phytoalexins are non-specific to the pathogens. Means phytoalexins act horizontally on each and every pathogen. They are non-selective to the pathogens. But pathogens are sometimes selective to the phytoalexins. Phytoalexins are not inherited. They can be produced in any plant species if it faces any type of stress or injury. Then phytoalexins are only produced in living moribund cells of the plant after attack of the pathogen. Phytoalexins are never produced either in dead cells or in healthy cells. Moribund cells means those cells surrounding to the dead or necrotic area, and these moribund cells are not healthy. In fact, they are weakened or attenuated due to the attack of the pathogen. Let me know some important facts about mode of action of phytoalexin on plant pathogenic fungi. First of all, degradation of cell wall of fungal pathogen. As we know that the cell wall of the fungal pathogens are made up of glycoproteins, then polysaccharides, chitosan, and glucans, and cell membrane. So all of these structures are disrupted and degraded due to when they are subjected to uh, the phytoalexin. Then second one is disturbed metabolism of the fungal pathogen. Actually, this phytoalexin causes the disorientation of the normal metabolic pathways. For example, ergosterol is one of the important constituent of the fungi. And and you can see here, this is the pathway to synthesize or biosynthesize ergosterol in fungi. So when this fungi comes into the contact of phytoalexins, the ergosterol biosynthesis pathway is altered or disturbed totally, and consequently there is no biosynthesis of ergosterol. Then we'll know about the mode of action of phytoalexin on plant pathogenic fungi. It is continued. So cytoplasmic granulation. Each and every eukaryotic cell have the cytoplasm, and when the cell comes in contact of phytoalexin, cytoplasm is disorganized or granulated. Cellular contents are disorganized or cellular organelles are disorganized. There is the rupture of a plasma membrane. Those fungal enzymes which are very essential for the spore germination, essential for different sugars, amino acids, and so many things. These fungal enzymes are deactivated when they comes into the contact of phytoalexin. If there is the spore of any fungal pathogen, if it comes in contact of phytoalexins, it does not germinate. It fails to germinate. Germ tube elongation is inhibited. Mycelial growth of the phytogenic pathogen is inhibited, and it prevents the reproduction, sexual or asexual reproduction in pathogenic fungi. Now, what is the site of production of phytoalexin in infected plant? Here you can see in this figure, this is healthy leaf. These leaf tissues are healthy, and these are moribund tissues. And third zone is dead and necrotic tissues with pathogen. So first one is, it is healthy tissues or healthy cells. So in healthy tissues, no phytoalexins are produced. This region, in this region, phytoalexins are not produced. In moribund cells, phytoalexins are produced abundantly and profusely. Then in dead and necrotic area, phytoalexins are not produced, but phytoalexins are diffused from these moribund tissues to the dead and necrotic tissues. These phytoalexins, when come to the contact of the pathogen or pathogenic fungi in dead and necrotic area, these completely kill the fungi. Well, so let we say about the quantity and duration of phytoalexin produced in plants. One of the example of bean plant, where bean was challenged with a bacteria, that is Pseudomonas syringipathy variety of Fasciolicola. So at the y-axis there is the phaseolin concentration in micrograms per gram fresh weight, and at x-axis there is the duration after the inoculation. We can see that after challenging the host with the pathogen, there is the production of uh, phytoalexins, but it reaches at the peak after 4 days of inoculation, and after that either the production of phytoalexin remains static or there is the decline in production. So what happens if the biosynthesis of phytoalexin is inhibited? Actually, if any host plant, after the attack of any pathogen, maybe bacteria or fungi, if the uh, biosynthesis of phytoalexin is inhibited, it becomes susceptible to the pathogen. So susceptibility of the host depends on the production of phytoalexins. You know that virulence of the pathogen is relative to its capability to degrade phytoalexins. Those pathogens who cannot degrade these phytoalexins, they prove themselves as a poor pathogens. They actually don't possess very high degree of virulence, but those uh, those pathogens which can detoxify or degrade phytoalexin, their virulence is at high level. You know, wild type of the host are more resistant to the infection. Wild type of hosts are more resistant to the infection because in wild type host there is the abundant production of phytoalexins. Now question comes, what are the biosynthetic pathways which are followed by uh, plant pathogenic fungi? So two types of the pathways are followed for the production for phytoalexin. First one is the cyclic acid pathway. Through this pathway, flavonoids phytoalexins are synthesized. For example, cavetone and phaseolin from bean and pyzetin from pea and glycerolin from soybean. And second one is mevalonic acid pathway. The gossypol in cotton, casebean in castor bean, and ricin from the Solanaceae family, particularly potato. These are known to synthesize through the mevalonic acid pathway. And these are the molecular structures of some representative phytoalexins, pyzetin, then 6-methoxymalin. So these are molecular structures. And now we'll know about the distribution of these phytoalexins in different families of the plants. So phytoalexins are especially widespread in family Fabaceae or Leguminosae and Solanaceae. Besides these two families, phytoalexins are reported in 15 other families also. Phytoalexins are not cosmopolitan. They have not been reported from family Cucurbitaceae and in Poaceae, Gramineaceae family, though there are a few of the examples. For example, in oat, in sorghum and rice, these phytoalexins are reported to produce, but apparently these are not produced in maize. So this is the distribution of phytoalexin in different plants. You can see here that these phytoalexins are mainly of two types, sesquiterpenite and isoflavonoid. Under sesquiterpenites, other category is diterpenites. So mostly these terpenites and isoflavonoids These are type of the compounds found in phytoalexins. So here in potato, pea, tomato, grape, beet, and rice and French bean. So these phytoalexins are produced by different crops. Thank you, dear students. Please like, subscribe, and share my video. Thank you.